Pattern generation for muscle movement. The center pattern generator, CPG, is a neural mechanism that generates rhythmic motor patterns such as walking, swimming, and chewing, without the need for conscious input from the brain. It is a network of neurons that produce complex coordinated movements without requiring any feedback from sensory systems. The CPG is found in animals, including humans, and is located in various parts of the nervous system depending on the type of movement. For example, the CPG for walking is located in the spinal cord, while the CPG for chewing is located in the brain stem. It is considered autogenic, self-regulating, self-generating, and involves a lot of complex coordination. Motor pathways and muscle tone and posture. Muscle spindles help monitor muscle tone and posture. Sensory receptors found in the muscles and tendons play a critical role in regulating movement and maintaining posture. Muscle spindles are specialized muscle fibers that detect changes in muscle length and the rate of change in length. They are located in the belly of a muscle and are connected to both muscle fibers and sensory neurons. When a muscle is stretched, the spindles are stretched as well, triggering a reflex contraction of the muscle. This is also known as a stretch reflex. This reflex maintains muscle tone and prevents overstretching of the muscle. So they are stimulated by the change of the muscle tone. The sensation is sent to the spinal cord, where there's a reflexive action to prevent overstretching or stretching too fast. And the sensation can also be sent to the cerebellum for assessing that information. You can see right here that the muscle spindle is located within the belly of a muscle fiber. Within it, you'll see some of these intrafusial fibers. Connecting the muscle spindles is going to have the afferent sensory nerve fibers for picking up information, and then efferent is the alpha motor neuron and the gamma motor neurons. Golgi tendon organs, GTO, are sensory receptors located at the junction of muscle and tendon. They detect a change in muscle tension or force, and when the tension exceeds a certain threshold, they trigger a reflex relaxation known as an inverse stretch reflex. This is stimulated by tension, not by stretch. They're located within the tendon where the muscles and tendon meet, and these send information also to both the spinal cord and cerebellum. The function of this reflex is to protect the muscle and the tendon from damage due to excessive force. You can see the Golgi tendon organs right here, shown here, composed and linked with collagen. They send an afferent sensory neuron up all the way to the spinal cord, and that will then route to an alpha motor neuron to the muscle to allow it to relax. Disorders of the motor pathway. Motor pathway disorders in the upper motor neuron. They're generally caused by resulting damage or lesions to the upper motor neurons, mostly commonly found in the cerebral cortex. Signs and symptoms include muscle weakness to that area, decreased control over fine muscle movement, hypertonia, where there's muscle overactivity, leading to a decreased range of motion or difficulty in moving the arms or legs, spasticity, where the muscles will spasm without a signal and causing pain. One way to diagnose this is using the Babinski's test to stroke along the base of the foot towards the small toe to see if the toes curl down or extend upwards. And this can identify some of the upper motor neuron disorders. There are different types of paralysis. Quadriplegia is when there's a partial or total loss of all four limbs. As a result, there's usually C1 to C7 level of spinal cord damage. Hemiplegia is when there's a partial total loss to one side of the body. So the motor cortex on the other side of the brain, contralateral to the side of the body affected, is the one that's damaged. Paraplegia is when there's a partial or total loss movement in one or both of the lower limbs, usually damage to the T1 and below. And then apraxia is when you have damage, depending on the type of apraxia, there are various motor functions that are affected. For example, movements of the muscle to the face, or moving of the tongue, or even movement of the leg and arm could also be affected. Usually it's damage to the frontal and parietal lobes of the brain. Motor pathway disorders of the lower motor neuron. These are oftentimes caused by damage to the alpha motor neurons via trauma, viruses, autoimmune disorders, and general symptoms include paresis, which is partial loss or weakness in a part of the body, but not a complete loss. Atrophy, where the muscles begin to degrade and shrink down in size. Hypotonia, where there's a reduced muscle tone and lower activity, and the muscles feel very soft and floppy. Usually a negative Babinski's test can confirm if you might have lower motor neuron damage. If there's a positive Babinski test, that's upper motor neuron damage. There's also basal nuclei disorders, when there's damage to the basal nuclei in those structures like the caudate, putamen, amygdala, colostrum, pallidus. Some general symptoms include dyskinesia, where there's a group of involuntary motions of repetitive and unpredictable jerking motions like tremors, and even chorea, which is like a dance-like motion. Akinesia, where there's a loss or impairment of the ability to initiate the motions. And even hypokinesis, which is a reduction and decrease in voluntary movement or activity. Some of the common examples are Parkinson's and Huntington's disease. And last but not the least is we also have cerebellum disorders, where there's damage that's caused by damage to the cerebellum via autoimmune disorders, inflammation, tumors, and even poisons or toxins. General symptoms include loss of coordination, posture imbalances, difficulty with muscle movement and fluidity, hypotonia, and cerebellar tremor. Aging in the motor pathway. As we get older, the brain does change. Features include the cerebral cortex begins to thin out a little bit. There's also breakdown and deterioration of the pyramidal neurons in the primary motor cortex. There's also a reduced amount of dopamine receptors in the neurons of the striatum located inside the basal nuclei. And changes to the motor units because there's a decreased axonal transference, there's a reduced number of cells, mitochondria, and less muscle filaments. These can all negatively affect our motor pathways. As a result, you're going to have a decreased posture, balance, strength, and endurance. Keep in mind, one way to prevent this is to regularly exercise and train your body.